So we're going to talk about biological membranes. And biological membranes are just focusing on one part of the cell. Remember this part of the cell that is the boundary or border of the cell. And we're going to talk in this chapter about its structure, the various parts that make up the membrane, and then its primary function, which is transport. So we'll start by looking at the structure. The basic framework of a membrane is a phospholipid bilayer. Bilayer means two. So we're talking about two layers of phospholipids. And just to refresh your memory, a phospholipid was an example of a lipid that we talked about in chapter 3. And at that time, we kind of kept it really simple. The reason why, or what's special about it, is that it has a polar head group and then nonpolar fatty acid tails. Because this part is polar, it's hydrophilic. Okay, and because this part is nonpolar, it's hydrophobic. So the molecule, a phospholipid, is amphipathic. That's again this term that we saw in a previous chapter, meaning both water-fearing and water-loving. So this is a diagram of the membrane. This is one layer, also called a leaflet. This is the second layer. So there's where that term bilayer comes from. This is the polar head group with the little fatty acid tails. So I'm just putting on top of here my more simple representation of a phospholipid. So there's one layer, and here's a second layer. So again, just putting my more simple diagram of a phospholipid here. And so what you can hopefully see is that this top part is hydrophilic, and this bottom part is also hydrophilic. But on both layers, the fatty acid tails are pointing in, so this whole region is hydrophobic. The reason why these have to face this way, where the hydrophobic part is always facing in, is because there's water on either side of the membrane. So the inside and the outside of the cell are based in water. So parts that like water, the hydrophilic parts, will always face towards water, and these will want to be away from water, and so they'll have to face in. So if you're looking at this diagram, which part is the most significant part of the membrane size-wise? So just thinking about this diagram, hopefully you can see that the largest portion of the membrane is hydrophobic. So we'll see in a little bit that this is really important. The membrane is really a hydrophobic barrier. This is a big hydrophobic region with just a little skin of hydrophilic on the top, but the majority of the membrane is hydrophobic. So as I said, this phospholipid bilayer is the primary framework of it, but we'll also see that there are proteins and carbohydrates. So there's three components to the membrane: the phospholipids, which is the phospholipid bilayer, proteins, or membrane proteins, and carbohydrates on the membrane. And we'll come back and talk about these two in more detail in a little bit. So what we understand about the membrane is summarized in this idea of the fluid mosaic model. The fluid mosaic model has two parts to it, captured by the name. The first part is fluid, which is that the membrane is a semi-fluid. This is going to dictate some of its behavior. It's almost more jelly-like. If it wasn't a little bit hard, it wouldn't be able to be a boundary. But because it's fluid, it's going to allow materials to pass through. So I guess probably the more important thing about it is it's not solid. If it was solid, it would totally block things from passing. Because it's not solid, it's going to allow transport of materials. And we're going to be mostly talking about transport over this chapter. The second part of it, and the one we'll again, talk a little bit more about is the proteins and carbohydrates. And they create a mosaic. If you know what a mosaic is in art, this term will make a little bit of sense, or if you even just know about sort of the everyday use of mosaic, it's kind of like a mixture of, it means kind of like a mixture. And so this is a more a deeper look at the membrane. This is one layer. So this is a phospholipid layer here. This is another phospholipid layer. And so that creates the framework. If it's like a mosaic in art, that'd be like the concrete. And then studded into that, so stuck down into the phospholipids, you will see proteins and carbohydrates. So proteins and carbohydrates are embedded into the phospholipids and create a mosaic-like structure. So we can see the little studding of the proteins and carbohydrates in the membrane. They're there for a reason, and we'll talk about some of their functions in this chapter. Okay, so the proteins that are in the membrane are broadly classified into two categories. So we have the integral membrane proteins and the peripheral membrane proteins. And notice that the key word differences are integral and peripheral. Integral means integrated. So an integral membrane protein, like the one here, is actually embedded in some way into the membrane. Peripheral membrane proteins, on the other hand, are like peripheral vision, which means to the side of. So it's near the membrane, but it's not embedded in it, it's to the side of it. Integral membrane proteins, because they're actually embedded into the membrane, are going to have to span this hydrophobic region. So again, remember where we see these hydrophobic tails, that's a big hydrophobic area. So proteins that are embedded into the membrane are going to have large hydrophobic regions that span that hydrophobic area. So transmembrane proteins are like the one pictured here. And so again, notice that the protein has large hydrophobic area right here that allows it to do that. The other possibility, so these are kind of two versions of integral membrane proteins, is a protein with a lipid anchor. So here's a protein, there's a lipid anchor. So a lipid is hydrophobic, so that will be able to stick into that hydrophobic part of the membrane. So either strategy works to make a protein be integral, having hydrophobic amino acids or having a lipid holding it in there. Peripheral membrane proteins are like this one pictured here. It's hanging out near the membrane, but notice that it doesn't actually enter the membrane. So because it does not actually enter the membrane, it does not need a hydrophobic area. So it will somehow be near the membrane, but does not have to interact with the hydrophobic area. Okay, so thinking about these proteins, if you isolated a membrane protein and looked at its structure, and you noticed that it had a large hydrophobic area of amino acids, so let me just add here that what we mean by hydrophobic region is that it has lots of hydrophobic amino acids, because that's what proteins are made out of. So we've got a protein with lots of hydrophobic amino acids and then two smaller hydrophilic regions. So remember that would mean that there would be hydrophilic amino acids. And again, kind of referring back to chapter 3, that's how the amino acids are classified because it's a really significant trait about them. So what would you predict about this protein's location in the membrane? Well, remember we said that if there's a large hydrophobic region, this is usually embedded into the membrane. So this is probably an integral membrane protein. And this kind of example is a true science example. One of the ways that we predict whether a protein or a protein's location is to know whether it has large hydrophobic regions or not. So we've been talking about the fluid mosaic model, and we talked about the proteins, alluded to the carbohydrates, and we're going to talk a little more now about how the membrane is fluid. So again, remember it's semi-fluid, and the key is that it's not solid. So membranes can vary in fluidity because they are made out of phospholipids. So thinking back to what you know about fats, remember they're made of fatty acids, and remember we can have unsaturated or saturated fatty acids. Okay, we can have fats that are liquid, like oils. We can have fats that are solid, like animal fat. Okay, if you think about animal fat, it melts. So temperature affects fluidity of fats. So this is all hopefully things you know from your own experience, that temperature affects the fluidity of fats. So since membranes are primarily phospholipids, in the same way, they are influenced by temperature. So at higher temperatures, there will be increased fluidity. At lower temperatures, there will be decreased fluidity. So that is something that is a problem for organisms, and why homeostasis can be so important in some organisms. So in an organism like us, for example, we rely on maintenance of internal temperature. One of the things that happens when you have a fever is that the increase in temperature that happens when you have a fever is increasing the fluidity of your membranes. If your membranes get too fluid, they become so liquid that they just literally dissolve away and your cells break. Right? So if that membrane is too fluid, the cell will tend to break because it will just dissolve and wash away. On the other hand, if your temperature is too low, so if you were having hypothermia or something like that, the fluidity decreases, it becomes too solid, and then it can actually shatter. And if you've ever seen fat congeal, you know that if you stuck a spoon in it, it would crack. And so same thing can happen to your cell, just like that. If it's too cold, it'll crack, just like the fat on top of a pot. So fluidity, the correct amount of fluidity for an organism is very important. So besides temperature, the cell has its own way to control fluidity, and that is what is in the cell, sorry, in the membrane, the phospholipids that are in the membrane. So that's what we'll talk about here a little bit. So the factors that affect fluidity, we've already said that one of the external factors is temperature. Okay, these are internal factors, so things that the cell can control. So the length of fatty acid tails, the shorter they are, the more fluid. So less length, more fluid. Okay, so less length, more fluidity. Double bonds, so if I increase my double bonds, I will increase fluidity. And so remember, that's that unsaturated fatty acids. So if I make more unsaturated fatty acids, I will increase fluidity. And then the third one here, and this only applies to animals, is cholesterol. Cholesterol is like a fluidity buffer. And I know that you all have a bad opinion of cholesterol, but this is a really important role for cholesterol in our membranes. That's normal, physiological, and that we need some of all the time. And the reason why is because it stabilizes our membranes. So if there's high temps, then it will make it less fluid. If there's low temps, then it will make it more fluid. So if something's going on in the environment, it helps to stabilize the membrane in that environment so that our membranes are more less likely to be damaged. But again, it's only present in animals, and that's probably why you already know from nutrition that cholesterol is only found in animal products. Okay, so here's putting this information to use. Winter wheat has adaptations that allow it to survive low winter temperatures. One such adaptation involves maintaining the fluidity of its membranes. What might you predict about the phospholipids in winter wheat? Well, if you think about it, so low temperatures, whoops, hold on here. Low temperatures is going to cause a decrease in fluidity. So my membrane is going to be tending to be too solid. So for that reason, I need to increase fluidity. So there's two ways I can do that. Remember, I can have unsaturated fatty acids, so have more of those double bonds. That will increase my fluidity. I can also have longer fatty acid tails. So go from having a fatty acid tail that's this long to having one that's this long. That will also increase my fluidity. So those are all possible strategies that allow winter wheat to survive in the winter while most other, or many other, especially crops, die in the winter. Okay, last part of the membrane. So remember, we're talking about the fluid mosaic model. So we said that there's phospholipids. They are proteins in the membrane, and then the third component of a membrane is carbohydrates. So glycosylation is a word that means addition of sugars to another molecule. So it's the process of covalently attaching a carbohydrate to a protein or a lipid, and this would form either a glycolipid or a glycoprotein. So a sugar attached to lipid or a sugar attached to protein. And this is important because this is how you get this attached to the membrane. So the lipid or the protein attaches it to the membrane, and the sugar part sticks off of the membrane. So let me flip back here for a second to remind you of this image. So here it is. There's a carbohydrate sticking up off the membrane into the extracellular space. And here's another close-up of that. Here's a protein, and then there's a carbohydrate, which is in yellow, sticking off of the membrane. So the protein part is in the membrane, but the sugar part is sticking off of the membrane. So the role of these carbohydrates, one of the really significant roles in animals is as cell surface recognition molecules. So they help a cell know what is foreign and what is normal. We'll talk a lot about this example later on in the semester, but blood typing is entirely based on carbs on membranes. And they're the carbohydrates that stick off of your red blood cells. So that's what I mean about cell surface recognition. You probably know that blood type means that if you're type A, your body knows what type A blood looks like. If you're type B, your body knows what type B blood looks like. And if I tried to transfuse you with type A blood type, which was not your blood type, your immune system would recognize that that was not you, not your blood cells. So that's a really important role for those that we'll talk about again later in the semester. In other types of cells, there can be a thick layer of carbohydrates, and that's this here in black. And this thick layer of carbohydrates is often sort of a protective coat. It can help the cell from drying out and different things like that. Okay, so that's the fluid mosaic model. We'll stop here now.